Good morning, friends. Today we will talk about the general pathology of the fungal infections. These fungi are plant-like animal pathogens. Fungal cells are large and having thick cell wall, which is containing the ergosterol and polysaccharides. Fungal diseases, based on their capacity to infect the tissues, they are divided into the 5 groups. The only obligate parasites, those are called the dermatophytes. They live in the keratin layers of the skin and cause ringworm. Such infections are called the superficial mycosis. Group 2. Some fungi which establish themselves in the normal individuals, they are called as the systemic or deep mycosis. Examples are histoplasmosis, coccidiomycosis, blastomycosis, and sporotrichosis. Group 3. Subcutaneous mycosis or mycetomas. These are saprophytic fungi, enters through the skin and grows in the mycelial forms, and these cause localized lesions. Group 4. These fungi cause both localized and disseminated diseases. Examples are the aspergillosis and zygomycosis. These are more common in the debilitated animals. Group 5. These are yeast-like single-celled fungi. These are not highly pathogenic. Examples are candidiasis and cryptococcosis produces the superficial infections, and very rarely they cause the systemic diseases. These fungi within the tissues, they produce the lesions. These lesions, they can be stained by the hematoxylin and eosin, or with the special stains like gomori-methenamine silver, periodic acid stiff reagent, or gridley stain. Presence of homogeneous brightly eosinophilic material known as splendor-Hopley material, which is surrounding the individual organism or colonies of organism, it is a characteristic feature of the fungal infections. It is believed that this material is produced by the antigen-antibody complex. These lesions, they are called as the classical granulomas, which consisting of the central core of necrotic debris, usually containing the pus. It is surrounded by a zone of epithelial cells, macrophages, and multinucleated giant cells. Variable number of lymphocytes, plasma cells, and also sometimes eosinophils may be present. Superficial mycosis, they do not produce the granulomas, only restricted infection to the skin or superficial layers. Here we can see within the figure classical granuloma within the center, which contains the splendor Hopley material, which is showing as a bright pink color colonies surrounded by the giant cells, that is surrounded by the eosinophils and also the lymphocytes. The defense against the fungal diseases, it is made by the alternate pathway of the complement system. Very small fungi, they can be ingested by the macrophages or the natural killer cells. The immunity against the fungal infections, those can be made by the T-cell mediated mechanisms. The fungal infections can be destroyed by T-cell mediated mechanisms. Some of the T-cells and NK cells, they cause the indirect cytotoxic effect on the yeast cells such as Candida albicans and Cryptococcus neoformis. Here within the figure you can see ringworm infection, that is the superficial mycosis infection. Second one is the aspergillosis within the guttural pouch of the horse, which will cause the granulomas. Example of the deep or systemic mycosis. Here we can see the classical granuloma in the intestines of the fungal infection. Within the center you can see splendor Hopley material surrounded by the epithelial cells and giant cells. The last one is the polyp, which is example of the subcutaneous mycosis. All these fungal infections, they are most common in the animal populations. These fungal infections, they are classified into the 5 groups. Lesions seen in the superficial or deep tissues. Defense via macrophages or T-cell mediated immunity. Fungal infections are the treatable infections but long-lasting infections. Here within the slide you can see references. Thank you.